could you just explain to the audience what you had done and what you found out?
Sure. Well, first of all, I did this at a flyaway executive jet airport in Jacksonville, where kind of people fly in, and general aviation, but they fly in and they're mainly testing pilots' hearts. Because I guess you can't fly to Davos to go talk about global warming unless you fly in on a jet and, you know, to hear Greta talk. So that people want to have unvaccinated, they want unvaccinated pilots. I'm not making this up. And this Josh Yoder that ran this was actually getting calls for unvaccinated pilots. But anyway, I'll just show you this really quickly. So there's 2 leads, one on each ankle, and then there's 2 leads, one on each wrist, and then one on your heart. And then you get this report. After 10 minutes, they do 5 82-second tests, because that's apparently the length of time it takes your blood to circulate, 1 complete cycle. And you get 5 readouts. You get 5 readouts like this, which just making sure that the test is good, the leads are picking up the signal, and then you get a severity number here, and whether the symptoms are local or global. So the severity here is you want to be 0, and the scale goes up to 22. But you'll see, so that's the general, and then on the back is where the juice is. You get.
Wait, can I just interject one? Is
Sure.
This at you're just laying down, you're just sitting, or do they actually stress the heart at all?
Laying down, and you're not sleeping, but you're not talking or anything. And so on the back, they give you the pathological conditions, and there's 12 of them, and then there's another 12, and I'll turn it sideways so you can read it. It's going to be hard to read, but you can sort of read those, sort of. And there's negatives or zeros is good, and then if you get a plus, that's where you're getting a hit. And the 3 different myocarditis right there, you can see all 5 tests, and then 4 tests on I get hits on all those. And so they're talking about fasting glucose and fasting insulin and change my diet and you know, all those good things. But what I have noticed since getting the myocarditis diagnosis is there's this a doctor in London, Malhotra, Malhotra. This Malhotra guy, cardiologist, says that there's 28 times the likelihood of myocarditis from the vaccine as opposed to the virus, which I thought was interesting numbers, and I asked him for his evidence. But the cardiologist that I was with, Dr. Levy, in the Jacksonville fly-in, he said that he had never seen hardly any myocarditis in his 40-year career. And then all of a sudden, 2020 and 2022, just 21, just a straight spike up. 18,000 cases, I think, in 2020, 16,000 cases in 2022.
You remember the Israeli study that were saying that myocarditis is for SARS-CoV-2. Unvaccinated incidences are not increased relative to the control group. Remember that study that came out? Well, you know, I read that study when I had time, and the problem with that study is, my understanding is it was done in Israel. That's so, you know, it's not a full randomized, multiple ethnicity type test, to my understanding. I could be wrong on that, but you know, with my quick reading of the paper. In addition, they only checked it they only looked the follow-up for 6 months. So they're comparing a group that wasn't vaccinated, that didn't have COVID, versus a group that wasn't vaccinated that did have COVID. And they're basically saying that there's no statistical evidence to suggest that myocarditis or pericarditis increases with COVID. Well, if you're only looking at it for 6 months, you're not going to, of course, you're going to only see the same number of, smallest number of incidents between myocarditis and pericarditis relative to the control group, because you can go out far enough. The pericarditis and the myocarditis could be 12 months out or a month and a half. And again, they did it with only the D641G mutation. They didn't do it with Omicron. And more people have Omicron than had, than during the first wave. The waves of alpha, beta, gamma, delta, right?
Yeah.
So there's another dynamic there that I think that is happening with Omicron. So that's my first observation with when someone said, yeah, go ahead.
Well, the study that Steve Kirsch always talks about is there's two studies. One's a Taipei study and the other is a Thailand study. And he says, why aren't these studies being done in the United States before we mass vaccinate, which is a good point. But one has about 5,000, the cohort in Taipei. And after Dr. Campbell in England said this, so he's kind of turning around the corner against vaccines, these vaccines. He said 5% showed some kind of myocarditis marker after the first shot, but after the second shot, this I think is the mRNA, there's 17% of the markers. And then the smaller study, 300 in Taipei, was a pre and post. So they did a pre EKG, then a post EKG. This is supposedly third generation EKG that catches a whole bunch of things you don't get with a regular EKG, by the way. But so I got the best of the best test. But anyway, in this test, 29% of the young males that were pre and post tested had biomarkers for heart injury.
And this is really important that you're bringing this up, because again, when we step back and say, well, what's causing the myocarditis, pericarditis, or endocarditis? It could be inflammatory response because of the spike protein, certain types of concentrations of the spike protein. Or it could be antibodies to the different antibodies that are produced when the spike protein is presented to the MHC. Okay, two things could be happening, right? But when you bring up, and this is important, really important, that the markers increase after the second round, then during the second round of an inoculation, your IgGs skyrocket up. Your first shot, they're low. That's why they have to do a second shot. So this is moving more towards the idea that it's probably the antibodies that are producing the inflammation that's leading to the myocarditis. But it would be interesting to see if we saw messenger RNA versus protein vaccinations, like Novavax. All right, I suspect that the messenger RNA, because of the human line 1 integration and the realization that these messenger RNAs are producing more spike protein and lasting longer than they thought, that the messenger RNA sequence is lasting longer in the cytoplasm, longer than they thought, that there's probably leakage of spike, S1 subunit. And because there's leakage of this, it could be causing this inflammatory response. But as time goes down, and I'm thinking through this as I'm speaking, as time goes down and the messenger RNA starts to get degraded and it can't produce as much spike anymore, assuming human line 1 was not activated, then these individuals that may have been getting myocarditis inflammation, even though mild, it should abate and the heart starts to heal. So it seems to me that if it's chronic, if it's going towards that the myocarditis flares up and doesn't abate, then I think it's pointing more towards potentially the human line 1 integration. And there's the cause, there's this cause of repeated inflammation.
Yes.
On top of that, those antibodies that are now, when you talk to Cole, Dr. Cole, and he did the pathology slides on the heart.
Yeah.
Did he say that they saw spike
Yes.
protein, or did
Well, they
or
didn't
did he
see,
say did he
he didn't say
see
spike protein.
IgG?
He said spike protein damage, and he said lymphocyte. And he made sure I said eosinophil. He said no, lymphocyte. So you know, it's that to me says IgG something. And you know, just to wind the
So,
clock.
so he's coming from the point of view that it's both. There seems to be when they do those stains and they do those immune,
Yeah.
immune stainings, or or some sort of fluorescent staining, or ELISA. He was seeing in that tissue spike protein and
Well,
antibodies
he
attached.
wasn't seeing the spike protein because you can't see that with a light microscope, but
No. But if they let's say
stained.
they did an ELISA test where they had an antibody to spike, that it would flare up. That they would, through
Yes, through the conjugate of
association, you would see that it's there. Okay,
yeah.
so it's looking like that. So it's looking like it's both now.
Well, when you
Go ahead.
said, just to stop you on human line one integration, because if people don't remember this, Paul and I had some discussions like February, March 2020, 2.5 years ago. And Paul theorized that the danger of the spike or the subunit was that it could bind to other things other than the ACE2 receptor. And one of the places was the heart. It was CD147. And Ryan Cole confirmed that. For less than a week ago, I heard him say that this is the CD1, where this slide was being stained. He said this is CD147. So he's confirmed what you said 2.5 years ago. So the other thing is the human line one, because you also said there was an affinity on the T cell. I don't know if it was CD147 or DPP4 or what it was, but that it could infect the helper T cell, and then the helper T cell, in trying to come up with an inflammatory response, has read this mRNA into its DNA and now is making spike proteins like crazy forever. Right.
Well,
That's
right there. What's happening is that, and this this is there's two categories here. There is the category where there is an immune suppression, and then there's a category for certain people where there's a dysregulation, where Treg isn't working correctly, and there's an overproduction. There's an overreaction of your immune system. All right. So for the areas of inflammation that is activating CD147, that's most likely not an autoimmune issue. It's most likely specifically a spike protein issue or an S1 subunit issue. So that's leading if more of these cases are because of this activation of CD147, then it is it's a spike protein activation. It's a direct spike protein activation. So higher viral load, more chances of myocarditis. Higher S1 subunit load, higher chances of myocarditis.
Oh, sorry. I'll show you the slide and healthy tissue. And I don't know, it says normal heart muscles and lymphocytes invading, if you want to put me on full screen or whatnot, but
there.
Yeah, I think maybe when I'm speaking, it puts you on full screen, but you can see the normal heart tissue with the no blue stains, and then the blue circular little dots there staining with the heart muscle damage. And then he showed several slides, expression of spike protein in the deltoid muscle, right? And saying it was getting into the bloodstream. And then once it gets into the bloodstream, it's kind of like metastasis. It can go anywhere. And it's kind of like it. Sorry, it's all of a sudden playing that slide. But you get the idea that he was saying the spike protein is causing all this damage. very.
And that would explain
Here,
the reason.
I'll just play in a
Right.
little bit.
That would explain the reason why for the. All right, go ahead.
The one who's being lungs, your kidneys, etc. That's fine, but they need to remember you are a whole body. So on that note, I'm going to go into a different realm. My name is Ryan Pohl. I'm a board certified anatomic and clinical pathologist trained at the Mayo Clinic. Been in practice for 20 years privately. Been in pathology for 26 years. I've seen 500,000 patients personally myself with a microscope in my career. So I have a little bit of experience. What I want to talk about real quick, I'm not here to judge anybody. If you got a shot, fine. If you didn't, great. If you got one, don't get two. If you got two, don't get three. If you got three, don't get four. If you haven't gotten one, never get one. And I'm going to explain why really quickly, because the cells don't lie. The human cells don't lie. As a pathologist, this is what I do all day long. I look at a part of you. I'm the most important doctor you never meet that you always hope is right. And pathology is the quality control of all of medicine. Treatment doesn't happen until those lab results are there. And then your doctor can go, oh, what do we need to do? What do we need to adapt, etc. So we're going to go quickly through the slides.
So, and that's where he starts into his slide pitch.
Right,
And-
right. So it's seeming like from this conversation that more often than not, it's the spike from the S1 subunit or the spike from the virus that is activating the inflammation, probably through CD147, but I suspect others. And that inflammation may not abate because of this dysregulation with Treg. Now, we step back and it's like, you got to look at the mosaic. And I think this is where what you and I do really well compared to Cole or McAuliffe or even Judy. All right. So the problem comes in is it's like, if anyone has played chess and you're almost in checkmate, right? You can't really move your pieces very well. So when incidences of cancer are going up because of the NSPs and the ORFs, which are not in the vaccines, but only in the virus, and severity of infection does go down with vaccines, then to say not to have vaccines to prevent myocarditis is maybe not prudent. I'm not pro-vax at all. But the point I'm making is that whatever choice you make, there's a problem. You pick the vaccine and you're probably going down the road of some sort of autoimmune or attack of your heart, the blood clotting issue, vasculitis, lots of other things that could happen, right? If the integration happens, it's like a fragment bomb and you don't know where it goes into your genome. And if it happens to disrupt DNA repair mechanisms, bam, cancer. Same thing with the virus. It may not elicit myocarditis as often. We don't know because we don't have a long enough study to prove it. But with the integration of a larger genome, and that's a larger fragment bomb, there's more chances of disruption of the chromosome, more chances of turning on and off because of this epigenetics thing through NSP 5 and 8, tumor suppressors and oncogenes disrupting that epigenetics that leads to cancer, right? So no matter what you pick, doctors need to come to the realization, just like doctors didn't come to the realization 3 years ago that we had a problem, that this was not zoonotic, that this was man-made and it was like from hell, right? Whatever you pick is gonna be problematic. And it's about risk assessment. It's almost like the only ones that really understand this are the ones that are like in finance. You have money, you don't know, you try to put your bets on, different companies, and you try to diversify, and you have certain risks for certain types of industries or sectors, and you work out some sort of complicated risk analysis, and then you place your make your portfolio. That's where we're at with this. So people on Twitter and on Facebook and even on YouTube and all this, they're thinking that there's an easy path. There's one path, and that because they picked that path, then they're okay. And it's the reality is that no one's okay, because there's no path that's okay, that's correct, because there's risks everywhere.
Yeah, I totally agree with you, because I'm with you. I don't say it tastes great or less filling. I'm not saying it's virus. I'm not saying it's vaccine. I think both spikes or subunits cause this thing. Yeah. But what I do think is that these weapons engineers, when they were all at the Soviet Union, and then they all got moved to Odessa in 2005 with the BioShield Act and the PrEP Act, and they all started coming to America, to American universities, and working at places like University of Florida and this Bob Malone company called Alchem. I look at what they specialize in. And when they were designing bioweapons all the way back in Russia, and they were not going for the stalk of the head of the spike. They were going for the stalk, like you said. They were going for this FC pathway. They were going to go for the functional constant, the FC pathway, because it was the same for everything. No matter what they threw in defense on the outer defenses, they were going to submarine right into the FC pathway and cause antibody dependent enhancement, which I don't know why they do that, but that's affecting the T cell. That's the thing, the helper cells that are greatly divided, and your body makes billions of when you're sick. So you're actually, if they can get that DNA, that RNA messenger into the DNA through some kind of retrovirus vector. And there was this guy named Cottrell in February 2020 who said, hey, why do they have a retrovirus in here? Why do they have the GP120 to get it into a cell and into the DNA of the cell? Once they get it in, it keeps replicating long after the virus is cleared. That's my point here. When you were saying the human cell line, that's what you're talking about, is it's becoming immortal. For the rest of your life, you're going to have these T cells with this implant. Is that right? Isn't that right?
Yeah. Yes. Yeah. And the thing is that if we step back and say that this is happening with the virus, and this is happening with the vaccine, the S1 subunit, okay, there are more issues when that happens from the virus than from the vaccine. Now, again, like I said, no path is free and clear. Both paths are catastrophic, all right? But they're catastrophic for different reasons. There's the devil's in the details, right? If you don't get vaccinated, certain people can have really bad infection. They could die from it, right? Especially if they didn't have any prior, infection, right? And if someone took the vaccine, they may not have as much of this. If someone did take the vaccine and didn't have the severity, but they could have this, these other problems, the myocarditis, even without the integration, or with the integration, and it's never ending. You can't get it out. This is the point I've been making, is that once it's integrated in, once it's disrupted your genetics, it could be producing that protein forever, whenever that region is activated to code for, or even more dangerous, if it disrupts the DNA repair mechanism. Because when that cell is starting to divide, it's starting to break apart. It's starting to turn cancerous. And it's the same problem with the study with the Israelis. The longitudinal study didn't go far enough. The follow-up didn't go far enough. 6 months is not a good way to compare myocarditis after COVID versus a control group that didn't have COVID, all right? You need to go out 12, 15 months, and most likely you'll start to see the signs,
Because
same thing with cancer.
if they're reading stuff into the T cell genome, right? Then another hit after you have the vaccine. You and I both had the J&J, and so that was a subunit vaccine, and so we're taking it in, okay? And supposedly that didn't affect our genome, but if it somehow got to, they used the FC pathway and they got to our genome, and now we're creating spikes every time we encounter something that inflames our immune system and alerts our immune system. Now we start replicating these T cells, and maybe we're making spikes, we don't know. And that's what's causing, so subsequent infections with variants that are slightly different causes a new activation. And now every time that happens, we're releasing more T cells, because I had no problem a year and a half ago when I got the vaccine, but then I got COVID. And then I started feeling a little bit more shortness of breath. And now that the second time I got COVID, now it really, that's when I went in for the test. So this idea of the second, third, and fourth infection, where you think, hey, I'm building natural antibodies, but in actual fact, you may have had something read into your T cells and you're making spikes.
Mm-hmm.
Because Dr. McCullough talked about a MHC study where he talked about the variable region, you know, the part of the MHC that configures the variable region being somehow like smashing the fingers of the piano player. They lose the dexterity of making the appropriate antibody. So that was another thing that they said, maybe there is uptake into the DNA, and it is changing some of the DNA with the MHC genes.
I think it is coming. I think there's a mosaic of data points that are starting to pop up. You know, it's in the early phase. It's kind of like the sun is starting to set and you're starting to see the lights in the city start to turn on, right? And they kind of
I don't like those lights very much.
but the point that I'm making is that more and more of these data points are popping up that are suggesting that there's integration. And I think that it's important to there's two main themes of the integration. The nucleotide sequence is fully integrated into a region that's coded, or this thing fragments, which it can, because it gets degraded, but because of you don't know, this can be like a fragment bomb, and the fragments of it get integrated in different areas. And so there's two dynamics here. When it's fragmented, there is actually a different type of damage. That's more of the cancer kind of damage that I'm talking about. When you have a full nucleotide sequence that's being integrated, and it's in a coded region, then that is okay, we're going to make more spike protein, or we're going to make more NSP5, right? See, this is when people start realizing that it's oh my God, this thing, and that integration is not just the vaccine. That integration is through the virus. That's why I keep on saying, people, maybe I speak hyperbolically when I say we're doomed, because I want to wake up people. You got to kind of wake them up so they can realize that something's happening. But, integration isn't just with the vaccine. The integration is happening with the virus. And, that we need to wake up here now, and the way it integrates, this is, I think, a point that you're making very well here. If the integration is in a precursor cell, such as an immune cell, then that's a different dynamic than an integration in, let's say, in some sort of crypt cell in the colon. See, so the pathology is different when things start to go wrong. See, if you have the integration and you start to have problems in the crypt, in the colon, then you're going to start seeing more polyps. You're going to see knocking out of tumor suppressor pathways that leads to cancer, versus integration in a precursor cell in your bone marrow, right, or in your lymphocytes. that's a different presentation, because now it's dealing with your immune system, or your erythrocytes, or your platelets, right? So this thing will present differently. I think with one of the professors that did the grand rounds, I told you 2 years ago, 2.5 years ago, he said, students, SARS-CoV-2, COVID-19 is going to mint like 20,000 or 20,000 new PhDs, and this thing's going to be with us for 30 years. And that this thing's so complicated, so complex, that we're not going to fully understand it. And as we move towards, we're in the third year now, that prediction seems to be holding still. This thing is a very complicated problem. And so it's hard to kind of like digest that, George, in, a 160 or 240 character tweet, to try to get people to like wake up.
No,
because
I'm
these camps, man, they just like, I didn't take the vaccine, so I'm okay. It's like, huh? Or the other side, the other side goes, I took the vaccine, so I'm okay. It's like, huh? You guys aren't paying attention to what a lot of people are saying. It's like, warning, warning. We have a problem on all dimensions here.
Yeah. Well, I look at it, I sometimes think of it like I'm a bioweapons engineer. Let's say I was working in Koltsova, like Michael Callahan, and I then got a good job down in Odessa, and I moved all my lab full of pathogens down to Odessa, and now I'm making some good money. And then they gave me a promotion. Now I'm in Alachua, Florida, by the university. I'm working for Allchem. I'm making some really good coin. And they say, well, I want you to design the bioweapon of all time. They say, well, we'd always use the FC pathway. We would always go for the T cell. Why? Because I can't get to the bone marrow yet with PEG. With a PEG coating, I can get through the bloodstream, but I get stopped before I'll have uptake to the bone marrow where the red blood cells are formed. But I can get to the spleen, and I can get to the thymus through the lymph system. That's the easiest attack route. It's sort of like the old Nazi scientists went for the spinal column because there was no immune system.
This is why I keep on saying that this crisis is in tandem with HIV. There is a HIV component to this arc of development. It's about
Absolutely.
immune system.
HIV was the FC pathway. That was the pathway to say, as the immune reaction occurs and the activation occurs, it'll make copies of the thing I want it to make. And the harder your immune system tries to stop the one, it makes the mutant, if you will, that just totally pollutes your body. Your body is working against itself because it's actually making copies of the thing that's going to kill you. And that's why you need the cocktail. That's why the cocktails work, because they attack 2 or 3 different things at once, right? They attack not only the primary, but then they attack the secondary, I'll call it, right? And the cocktail turned out to be why everybody's living right now. You said this in February of 2020, it's like, oh boy, I see these HIV inserts, I see the retrovirus inserts. It sounds like there's going to be.
And the people that were doing it were
Yes.
tied to HIV research,
Bob
directly
Malone,
or indirectly.
Robert Malone goes all the way back to Murray Gardner's lab, where Murray Gardner is the guy who took the HIV virus from Luc Montagnier in Paris and flew back to California, and he started the monkey lab in University of California.
I didn't know that.
Yeah, Murray Gardner. Yeah, and Bob Malone worked for Murray Gardner, and then they took that down to Salk, and then at Salk, Bob Malone was adding GP120 to everything that because he couldn't get uptake. He was trying to infect all these different kinds of cells, and he couldn't get uptake. So he was tagging. They would put a poly, a quad poly tag on the end of all these molecules, adenosine, 4 adenosines in a row, and then slap a GP120 on the end.
Do you know what year he did that?
87.
Okay, so this is interesting. Something that I didn't know that. So, of course, when we have these conversations, a whole bunch of new stuff comes out of my brain. So there's a body of literature because of these researchers doing what they were doing that are starting to happen in late 80s and early 90s that leads to evidence to point to create the pseudovirus that Dr. Shi did in 2007. Because you got to remember, SARS didn't have a good take.
Right.
That was developed in 2001. Didn't have a good take, right? But the take was really good once they took the research from Shi with that HIV homology.
Right. Right.
So there's this
Because it's
buildup.
kind of,
There's this. I didn't know Malone was part of that buildup of why glycoprotein 120 was working.
he went to
See, I
Viking.
thought Malone was just okay. He was just a synthetic kind of messenger RNA guy. I didn't
No.
realize that he had HIV
Big time.
experience.
Big time. And
Wow.
at Vical, when he was at Vical, and Dr. Verma, he said he got PTSD from Dr. Verma because he was an HIV researcher. And and Verma said Bob Malone was stealing all of his stuff, and there was big. But then there was this guy named Wolf, John Wolf, who also did.